Hello, guys. So what is it, Wednesday, October 21st. How's the connection? Can you hear me?
Yes.
Thank you. So we are going to start talking about chapter 8, nucleic acids. So we have covered so far different kinds of biological molecules, macromolecules. So we have covered proteins. So we talked about amino acids as the main constituents of proteins. We talked about carbohydrates, which have monosaccharides as the main constituents, right? So for example, glucose can undergo polymerization to make polysaccharides such as starch or glycogen. So we're gonna start, so today we're starting a new type of biological macromolecule, nucleic acids. So we'll learn that nucleotides are the main components of nucleic acids. So the homework was due this morning. I opened the next homework for you. And so we have 4 lectures on this new topic. 4 lectures, and what's today? Wednesday, Friday, Monday, next Wednesday, and then Friday before Halloween, we have next test. And let me know if you have any questions about the material or any other questions.
Quick question. Did you say the test is on the Friday before Halloween? I think
I
had down it's on Monday.
I'm sorry, I gave you the wrong dates. These are for my other class. These are for my other class. So right. So on Friday we don't have a lecture. That's where I messed up. So we have actually 3 lectures, not 4 lectures. So I take it back. So today, Wednesday, then Monday, Wednesday of next week, and Monday after Halloween, right? That's when November 2nd. That's when we have our second test. Well, just make sure you have your syllabus handy, right? Put the syllabus on your nightstand so that you know. Make sure you don't miss anything, even though I'll be reminding you about everything through Canvas, of course, but just in case. All right. So share screen. And we have our nucleotides and nucleic acids. So we will start by discussing biological function of nucleotides and nucleic acids, right? So specifically what they do in biological systems. And from that, we will talk about their structures. We will start with common nucleotides. As I mentioned to you, nucleotides are constituents of nucleic acids, right? So we'll start by discussing structure of nucleotides, just like with proteins. So we started by discussing structures of amino acids. Here we're going to start by discussing structure of nucleotides, and then we're going to put them together into nucleic acids. And specifically, we'll talk about DNA, deoxyribonucleic acid, right? And we'll talk about how the DNA forms, how the nucleotides form base pairs, and specifically double-stranded DNA. We'll talk about ribonucleic acids, RNA as well, right? Talk about RNA. So many of you already know the main difference is the structure of the ribose. One has hydroxy group, the other doesn't. So we'll look at that in detail as well. We'll talk about denaturation. So obviously you can what's called melt DNA. You can melt DNA to separate the 2 strands from each other. And so you can do this in the lab. Or you can imagine that this process occurs in our cells all the time. Whenever a cell divides, for example, when the genetic information needs to be read by the DNA replication machinery, right? When the two daughter cells are formed from a mother cell, and the DNA has to be copied, right? So the two strands have to be separated. The same is the situation when the proteins have to be synthesized, right? The genes have to be read through by separating the two strands and using each strand to convey the genetic information. And we'll talk about the chemistry of nucleic acids. So specifically, what kind of reactions nucleic acids can undergo. And finally, we'll finish by discussing mutagenesis, right? How the DNA, how the genes can be altered, which can give rise to mutated gene products and can give rise to various genetic diseases. And we'll discuss that in some detail as well. All right. So as far as the functions of nucleotides and nucleic acids. So obviously for DNA, the first thing that comes to mind is the storage of genetic information. So as we mentioned, to store something doesn't mean simply just to put it there and keep it there, right? So DNA and the whole genetic machinery has to work in such a way that this genetic information has to be accessed easily, right? So first of all, it has to be protected, obviously, right? It has to be protected from environmental effects, environmental stresses, so you don't get any kind of mutations in DNA and cancer and stuff like that. On the other hand, also, even though it has to be protected well, it has to be accessed easily, right? So you can imagine the cells, many cells undergo continuous cell division. And if that's the case, the DNA has to be replicated constantly. So storing the information is not going to do it. You have to be able to actually make it accessible for the enzymes, for example, DNA polymerases, enzymes which make copies of DNA. So these enzymes have to access the genetic information. And so storage is actually a big word, even though it sounds very simple, but in reality, it's actually a very highly sophisticated process, highly organized and regulated. Then transmission of genetic information into messenger RNA. So here we have RNA polymerases. So basically enzymes that copy the genes into RNA templates, right? RNA templates, which will then travel from the nucleus to the ribosomes, the place where the proteins are synthesized. But before it does that, the genetic information has to be processed. So, there is a term known as RNA splicing. Anybody knows what that means? If you've taken biology, you should know what that means. What's RNA splicing?
It's like translational or transcriptional control, I forget, but it's like they take out certain segments of the RNA so it can get read into different things or synthesizing different things, I guess.
Right, so there's introns and there's exons. Introns and exons. So exons are part of the RNA which will carry the genes. Introns are those that separate the genes, right? So the parts of DNAs have to be simply removed, and that's called RNA splicing. And in this case, actually, RNA behaves as an enzyme by itself, okay? And it actually performs its own kind of processing, right? Basically, it removes the parts, introns, and keeps the exons together before the mature RNA can go into the ribosomes and allow for the protein synthesis to occur. And so once that happens, then protein synthesis, so messenger RNAs arrives at the ribosomal RNA. So ribosomes is a large protein factories, right? The main component is RNA, but there are lots of what's known as factors. There are lots of proteins associated with the ribosome as well. And so that's the second main type of RNA. There is messenger RNA, ribosomal RNA, and there is, what's tRNA? What does that stand for?
Transfer RNA.
Transfer RNA. So what's the function of transfer RNA?
Is that the one that brings the amino acids to the ribosome?
Yes. So this tRNA, so these are relatively small structures compared with rRNA, for example. They will have their carriers. Their carriers, they will bring amino acid, and they will have the they will be complementary to messenger RNA so that they can bind to it and use messenger RNA as a template. And if the sequence of nucleotide matches, then the amino acid will be utilized in protein synthesis and will be incorporated just in the right place. All right. So these are main functions of DNA, and there are 3 types of RNA. Now, but besides that, so nucleotides are also used in the monomer form for different cellular functions. For example, energy for metabolism. So ATP, adenosine triphosphate, is the main molecule for, it's an energy currency in the cell. So for example, if you eat a meal, you digest it, and you convert those nutrients into energy. And how do you do that? You take adenosine diphosphate, you add inorganic phosphate, so that requires energy, and you make ATP. ATP, adenosine triphosphate. So the main component here is a nucleotide, the molecule based on the adenosine, which is the nucleoside, phosphorylated nucleoside. So we'll talk about nomenclature in a little later today. So enzyme cofactors. So remember, those are molecules. These are not amino acid based molecules, which are associated with enzymes to help enzymes perform their functions. So there is nicotine adenine dinucleotide. There's reduced form, NADH. NADH can reduce biological molecules and undergo oxidation in the process to NAD+. Right, to NAD+. And obviously it can go backwards. This is going to be the reduction of NAD+ into NADH. So the main function of NADH and NAD+ is the participation in various redox processes in the cell. And also nucleotides participate in signal transduction, for example, cyclic adenosine monophosphate. So these molecules are important components of signaling within the cell. And so we'll talk about that. I'll show you the structure of cyclic adenosine monophosphate. So this molecule binds to various enzymes and activates them or deactivates them. and thereby participates in signal transduction. All right, so these are functions of nucleotides and nucleic acids. Do you guys have any questions so far?
Yes, what did you say the N and an AD plus stands for?
Nicotine,
Okay, thank you.
like nicotine that in cigarette tobacco. And tobacco.
Got it.
Yeah. All right. So the main structure of nucleotides. So there are three components. There is a purine or pyrimidine base. So commonly referred to as nitrogenous base. There is a sugar, pentose, right? 5-carbon sugar, and there's a phosphate. So there are three components to nucleotide. So for the nitrogenous base, so nitrogenous bases are actually heterocycles. Remember the word heterocycle? Heterocycle is a ring in which some of the atoms are carbons, but not all, right? So you can see here in this ring, it looks like a benzene ring, but there are two nitrogens. And so this type of aromatic structure is known as pyrimidine. Pyrimidine, so like benzene, only two nitrogens. And the other one is purine. So purine, you can see here now it has two rings, right? They're fused at this position. One of the rings is actually pyrimidine ring. Again, 6-member chain with two nitrogens. And the other ring, anybody remembers the name of this other ring? So if you were just to name the 5-member chain here, what would be the name of this ring? Well, what amino acid? Remember you saw this in one of the amino acids. Which amino acid is that? So one of the amino acids has this structure of this 5-member chain with two nitrogens.
Is it proline?
No, proline is a good guess. It has one nitrogen, but proline, remember, is not an aromatic structure. And proline is a fully saturated, so it's not a heterocycle. It's not a, yeah. So what else? Histidine. Remember the name of the heterocycle in the histidine? The name of the heterocycle in the histidine is imidazole. Imidazole. So purine is the actually fused ring structure, which is a combination of a pyrimidine and imidazole. Now there is a specific numbering which is used for both pyrimidine and purine rings. Now based on their predominantly organic chemistry principles. So pyrimidine, you can see here, you start obviously on one of the nitrogens, right? So it has higher atomic number than carbon, one of the nitrogens, and then the second one, and you go towards the second nitrogen because that will allow you to minimize the numbers. If you go this way, then this nitrogen will be nitrogen 5, position 5. But if you go this way, it will be position 3. So pyrimidine, you start with on one nitrogen, and your other nitrogen will be N3, position N3. With the purine, the same thing. So here by convention, now I don't know if there are specific organic chemistry rules, but it was decided that you start numbering on a pyrimidine ring. And again, you start numbering one of the nitrogens. Let's say this is N1. And then you go towards the second nitrogen, N3. Then you complete your numbering system. Complete your numbering system. You end up with 6 positions, right? And then you go to the second ring and you finish the numbering 7, 8, 9. 7, 8, 9. So 9 total atoms in the purine ring system. All right, so these nitrogenous bases, they are linked to the ribose through one of the nitrogens. So in pyrimidine, this will be nitrogen 1. In a purine, this will be nitrogen 9. Nitrogen 9, that's how it's going to be linked to the ribose or deoxyribose. So what I also want you to understand is that in general, they're called nitrogenous bases. Why are they called nitrogenous bases? Well, they will have obviously NHs, so that they can be served as the hydrogen bond donors. But also remember, when something is a base, it doesn't necessarily have to be Bronsted base. It can be Lewis base. And remember, Lewis bases are those which have lone pairs of electrons. And since the nitrogenous bases have lots of nitrogens in them, that's why they're called bases. This one, right? And so because of that, in general, DNA, for example, or just in general, an RNA, one thing to keep in mind, DNA is nucleophilic. So why is it important? It's important when we discuss DNA damage. DNA damage. So DNA is usually damaged, and obviously if DNA is damaged, your genes can be messed up, right? You introduce genetic mutations, and then you get defective proteins and various diseases that follow, including cancer, right? So for example, this position N7 in the purines is highly nucleophilic, and it can undergo an SN2 reaction. Let's say you have a leaving group here, right? So this can attack it, and this will leave. So it's an SN2 reaction. And by doing so, you will make the nitrogen carbon bond, and obviously you will modify the DNA, right? And if you modify the DNA, then the genes may not be read correctly by the DNA polymer or by the DNA or RNA polymerases, which either form the proteins or lead to DNA replication. And so the mutations can be introduced. So such compounds, electrophilic compounds, are known as mutagens. Mutagens. And mutagens, in many cases, are electrophilic organic compounds, and they react with DNA because DNA is nucleophilic. And it's nucleophilic because of the lone pairs of electrons on the nitrogenous bases. All right, are there any questions about nitrogenous bases? So that's part of this substructure of the nucleotide. Then we move on to the ribose. move on to the ribose. So here, what do we have? Here we have so nitrogenous base. So there will be a nitrogen, obviously, right? Just like I showed you, and 1 of the pyrimidine, or N9 of the purine. And so this will be linked to carbon 1' of the ribose. And so this carbon is going to be anomeric. Anomeric carbon. And you can see the numbering. So the numbering, so this is C1, but to make this different from the bases, to avoid the confusion, right? To alleviate the confusion, I guess, we're going to be numbering ribose using these prime numbers, or using the designation prime. So it's going to be C1 prime, C2 prime, C3 prime, and so on and so forth. 4 prime and C5 prime. So here you can see the difference between DNA and RNA is the presence or absence of this hydroxy group, right? So in DNA, there is no hydroxy group. So deoxyribonucleic acid will be lacking the hydroxy group at position C2 prime. Ribonucleic acid will have the hydroxy group at position C2 prime. Now, as far as the anomeric position, now tell me if this is a beta anomer or alpha anomer. Remember anomers? How do you tell if it's beta or alpha?
Beta if it's cis, and alpha if it's trans. So I think beta.
Yes.
It has the CH2 up, and that's also pointing up.
Yep, perfect. Right. So, so with nucleic acids, the structure of nucleotides, they're beta anomers, because the nitrogenous bases are on the same side as the C5 prime carbon. Right. So what else do we need to tell about the ribose? We'll mention the Rinpacker in a little while, but the third main component is phosphate. The third main component is phosphate, and phosphate you can see here is mostly deprotonated. So the reason why it's deprotonated, you have to look at the pKa. Remember, so pKa, for example, if you have phosphate, let's say this one is deprotonated and this one is protonated. So the pKa of this is close to 0, right? And remember the physiological pH is about 7. So we have 7 units, right? 7 orders of magnitude. And so, which means it's that 7 zeros, right? 7 zeros is what, 10 million? 10 million, so which means that per 1 protonated molecule, you have 7 million deprotonated molecule of the phosphate. So phosphate is largely deprotonated. And so what that means is that DNA and RNA are largely not only nucleophilic, they're also negatively charged. DNA, RNA, are negatively charged. Okay. All right. Any other questions about nucleotides? Okay, so nucleosides, the difference, one thing to remember, the difference between nucleotides and nucleosides. So nucleotide will be a molecule with a phosphate. Nucleoside will be a molecule without the phosphate, right? Just the nitrogenous base and the ribose, deoxyribose or the ribose by itself. All right. So phosphate group, so as we mentioned, it's negatively charged at neutral pH and typically attached to 5' position. And their nucleic acids in general, they're built using the 5' triphosphate version of the nucleotide. So in other words, if you have a nucleotide, like so, here is the nitrogenous base, this is the ribose, here is the 5' position, oxygen, phosphate. So this will be nucleotide, right? But if you add 2 more phosphates, so this will be nucleoside triphosphate, right? So you have 1, 2, 3 phosphates. And when you build the nucleic acids, this will leave. Diphosphate will leave, and it's actually a great leaving group. So if you remember, great leaving groups are those that are weak bases, right? So remember the phosphate, the pKa is close to 0. So which means that it's a pretty weak base. Right, so this is diphosphate. So diphosphate is often called pyrophosphate. So this will be the leaving group, right? And then the next molecule of the ribose will be linked through this phosphate, okay? So we will find out if this is 5' position. This will be 3' position of the next ribose. And so this will be a weak base. Which means it's a good leaving group. Good leaving group. Right, so this will be adenosine, guanosine, adenosine triphosphate, guanosine triphosphate, cytidine triphosphate, and let me see if I get this right, thymidine triphosphate. Yeah, so with this nomenclature, we will actually look some more at the nomenclature. I still get confused about this quite often myself because, for example, nucleotides have a different name from nucleosides, and nucleosides have different name from nitrogenous bases. So, I mean, different suffixes. So you just have to use, you have to use this like in frequently to actually to remember those things. The problem is that my research group does not use nucleic acids in our projects. So I only go over this material when I teach this class, which is every two or 3 years. And I myself oftentimes have to remind myself about how to name these things. But what's important here is that these are triphosphates. Triphosphates, adenosine, guanosine, thymidine, and cytidine triphosphate. So 2 of these will leave because it's a great leaving group. And from triphosphate, you get the monophosphate. All right, so these phosphates can be attached to other positions as well. So as I mentioned to you, 5' is the key, 5' and 3'. 3' position, so this is adenosine, 5' monophosphate, and adenosine 3' monophosphate, right? So the nitrogenous base is adenine. Adenine, adenine. And here Now 2', so this is not very common. It's primarily has significance as a single nucleotide. So it's not really obviously part of the nucleic acids. But this cyclic one is part of the signal transduction pathways. So I was telling you about this molecule, right? So you can see here, position 3' and 2', they both carry this phosphate, right? It's almost like the way to see this is something like that, right? So when this hydroxyl attacks the phosphorus of the phosphate, and this will leave, obviously there'll be some kind of hydrogen bond donor to allow this to leave. And so important molecule in signal transduction. All right, any questions? Questions?
What's the purpose of the phosphate group being attached at different places again? I know you already said one of them was signal transduction, but
yeah,
what
2',
about the other
I'm
two?
not exactly sure. Maybe I should look this up. I mean, I have seen it before, but I don't know exactly which specific pathways it participates. But it's not part of the nucleic acids, so it has importance as a nucleotide by itself, not as part of nucleic acids. But 5' and 3', so these are nucleotides. So remember how, well, we will talk about how nucleic acids are built, but jumping a bit ahead. So if this is what, 3' position, right? And this is 5'. Then this is 3' again, right? So you can see there's this phosphate, right? So here and there, and there'll be another phosphate, obviously, here, right? Again, 3' will be linked to 5', and so on and so forth. So when you have a phosphate at 3' or 5', that's where it actually belongs, right? To build nucleic acids. Very significant. Now, sugar. So we talked about deoxyribose and ribose by itself. So the sugar is furanose, 5-carbon sugar. So this is the open Fischer projection. Important to realize all these hydroxy groups in the ribose are on the same side, on the right-hand side. If you make a ring, two of these will be down, and the 5' carbon will be up. Now this is beta anomer, but keep in mind can be also alpha anomer, right? I think there was a question in the discussion forum. I think there was a question when this cyclizes, how do we know it's beta or alpha anomer? And the answer is we don't know, because beta anomer and alpha anomer exist in an equilibrium with each other. So at any given time, you have alpha and beta when they're in the form of hemiacetal. When in the form of hemiacetal, then you have alpha and beta present in a mixture. So beta, alpha. Okay. They can go back and forth, and they can also equilibrate with the open form. So all these three forms equilibrate, and depending on the temperature, depending on the solvent, for example, you can put this in water, right? But then add some solvent, let's say methanol, you can shift the position of the equilibrium. For example, let's say you have hydrogen bond here between two OHs, right? Hydrogen bond, and water breaks up hydrogen bonds. But then you add some methanol, which doesn't break up hydrogen bonds so badly, and then you can shift the equilibrium towards beta anomer if you add methanol, because you sorry, what did I say? So water breaks up hydrogen bonds, and that it may favor beta. If you add methanol, you restore the hydrogen bond, right? And then you can form more of the alpha. Does it make sense? So the solvent, which either breaks up hydrogen bonds or facilitate hydrogen bonds, can actually shift the position of the equilibrium between beta and alpha. Now, also one other thing to keep in mind is that we draw these in the kind of, they look like planar, right? So if you look at the structure, looks like, oh, okay, it's a 5-member train and it's planar. But we know that the 5-member trains are not totally planar. They're actually puckered. They have this envelope conformation, and you can see what's happening here is there is this either a C2 prime carbon or C3 prime carbon. They can go sort of get out of the plane of the 5-member train and go up like so. And this can go up like so, right? And so remember from OChem 2, endo means same side, same side, exo means opposite sides, opposite sides. So so here the problem is obviously I'll go back again. The reason why this doesn't like the planar conformation is because of the steric hindrance, right? So remember eclipsing interactions, eclipsing. So for example, in 6-member train, remember chair conformation is the predominant one because it allows one to avoid eclipsing interactions. In the 5-member train, it's going to be the envelope conformation. And you can see what's happening here. As this C2 prime carbon goes up, then the base, instead of looking straight up, is starting to look away from the ring. So it basically kind of, as this ring goes up, right, then the base moves away from the ring and avoids, base means nitrogenous base. Base avoids steric hindrance. Base avoids stereohindrance. And the same thing happens here. So as this goes up, right, instead of looking straight up, the base is starting to point away from the ring and will avoid the stereohindrance as well. So these are called Packard conformations. Packard conformations. All right, any questions? So this is the ribose, not planar. Keep that in mind. And for the nitrogenous bases, so we looked at, so basically I told you that there are two kinds, or there are two main types. There's pyrimidine and there's purine, right? And so now these are heterocycles. And so the nitrogenous bases will be derivatives of these two. So these are heteroaromatic molecules. And so by themselves, pyrimidine and purine, they are planar because they're aromatic. Remember, aromatic structures like benzene or naphthalene or anthracene, all these are planar. And so are pyrimidine and purine. And also another important thing to keep in mind is that they absorb light in the UV region, right? So you can actually follow DNA or RNA, follow the chemistry or biology of DNA and RNA with the UV light because they show themselves in the UV region because of the aromaticity of the nitrogenous bases. And so there are 5 of these bases, right? So cytosine, adenine, and guanine, they found in both DNA and RNA. Thymine, only DNA. Uracil, found only in RNA. So all good hydrogen bond donors and acceptors, right? And neutral molecules at pH 7. Okay. So as far as purines, so we have 2 main kinds. There is adenine and there's guanine. And for pyrimidines, we have 3 kinds: cytosine, thymine, and uracil. And so this occurs only in DNA. This occurs only in RNA. So unfortunately, you have to remember these structures, guys. Now, again, you don't really need to memorize this from scratch. There are techniques and tricks that you can utilize to remember these structures. I would, for example, start with uracil. This is the easiest one, and it's component of RNA only. All right, so basically it's a pyrimidine with 2 carbonyl groups. You draw this very quickly. And then to go from an equivalent base in DNA, you just put a methyl group here. This is position 5. This is 1, 2, 3, 4, 5. So to go from uracil to thymine, you put a methyl group at position 5. Put a methyl group at position 5. So that's one thing you can do. Another thing you can do is if you look at the structure of cytosine and guanine. So here you have carbonyl on the bottom, NH2 on top, right? If you go to guanine, you have NH2 on the bottom and carbonyl on top. So they switch, right? Carbonyl bottom, NH2 top. Carbonyl top, NH2 bottom. And so, so for example, so then you can, knowing the structure of pyrimidine cytosine, you should be able to draw the structure of the purine guanine. Just switch to these 2 positions. Switch these 2 positions. But how do you know that it's guanine and cytosine? So what else is, besides this structural nuance, what else is common between guanine and cytosine? Like what else do you know about guanine and cytosine that that they share in common? like before coming to this class.
You just mean that they pair up?
Yes, 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 base pair. That's right. So, so you know that, for example, in DNA, right? We have two types, canonical, or sometimes called Watson-Crick base pairs: guanine cytosine and adenine thymine, right? And so G and C form a base pair. And so, and the reason why they form a base pair is because this is the hydrogen bond donor. This is the hydrogen bond acceptor. In cytosine, now this is hydrogen bond donor and this is hydrogen bond acceptor. So they switch their positions, and that's the reason why they pair up, as you said. Why they form a base pair is because you switch their positions. And this loves to bond to the central nitrogen on cytosine because this has a lone pair and this has an H, right? So we're going to look at the base pairs on Monday. But so that's one trick to remember guanine and cytosine in terms of remembering their structures. For adenine and thymine, so you know actually there are 3 hydrogen bonds here, right? And adenine and thymine have 2 hydrogen bonds. And the reason for that, if you look at the structure of adenine, there is nothing, there is no hydrogen bond acceptor or donor at this position. There is hydrogen bond acceptor here, there is hydrogen bond donor here, and so you can form 2 bonds, but you cannot form anything here, right? And so that's one way to remember, for example, the structure of adenine. Remember, it only forms 2 bonds in its base pair, and so this will be CH. This will not be hydrogen bond donor, not hydrogen bond acceptor. So you can draw this part, and so these are some tricks you can use. But overall, anyway, there are only 5 of them, so hopefully this will not present significant difficulty to remember these. So the nomenclature. So here's what I said to you, that sometimes I struggle with this thing. So so these will be nucleotides. So the bases by themselves. Nucleoside, remember this is the base and the ribose. Nucleotide, remember this is base, ribose, and a phosphate. And then which part, and then which nucleic acid this will belong to. So adenine. So this will be, remember, the nucleic nitrogenous base shown here, right? So it's a purine. Told you about the 2 hydrogen bonds. So if it's part of a nucleoside, has just the base and the ribose, so adenine becomes adenosine. And if the ribose is deoxyribose, then it's deoxyadenosine. Now you add a phosphate, and this becomes adenylate or deoxyadenylate. And so adenylate is part of RNA, deoxyadenylate is part of DNA. So this is one purine, and second purine is guanine. So this will form 3 bonds. So guanine, as part of the nucleoside, is guanosine. As part of the deoxyribose nucleoside, is going to be deoxyguanosine. Part of the nucleotide, again, this is phosphate, remember. So this is sugar, sorry, this is base plus sugar. Base plus ribose. This is just the base. Just base. Base plus ribose. Base plus ribose. Plus phosphate. So guanylate, deoxyguanylate. And then we'll have RNA and DNA. Now for pyrimidines, so cytosine. So as you add ribose to it, to the base, it becomes cytidine and deoxycytidine. If it's deoxyribose, cytidylate, deoxycytidylate, and that's RNA and that's DNA. And then uracil, thymine. So now remember, this is only the component of DNA. And remember thymine, the difference between uracil and thymine. Who remembers the difference? Without looking things up, what's the difference in structure? Methyl group. Position C5. Right, so if we go back, so remember difference uracil, RNA, thymine, DNA, and here we have CH, here we have C-methyl, right? So should be easy to remember. So thymidine, deoxythymidine, thymidylate, deoxythymidylate, DNA, and uridine derived from uracil, uridylate, and its component of RNA. Right, any questions? Well, I don't know if you can have a question here. It's just a nomenclature that we need to get used to. All right, so yeah, so you will need to know the structures, names, and symbols, both 2-letter and 4-letter codes. All right, so here is our nucleotide. Remember, nucleotides, this is deoxyadenylate. Now, there is a second name for it. So for the nucleotide, remember, nucleotide is the base, ribose, and phosphate. But you can also just name this part, which will be nucleoside, and then say that it's a monophosphate. So in other words, you can name this deoxyadenosine, which will be just the nucleoside, and then add 5' monophosphate. Right? It's just another name, another way to say to refer to a nucleotide. So deoxyadenylate, or if you forgot this adenylate, you can always use this term for nucleoside, deoxyadenosine, and then just add the phosphate. And why this is also useful is, for example, if you have 2 more phosphates, you can say that this is deoxyadenosine triphosphate, right? So symbols, now A. So A, sometimes it's used, even though this is primarily for ribonucleic acids, but when you primarily deal with DNA and you don't take into account RNA, some people just use A, right? So, so this is probably incorrect, but this is one of those things when this mistake is forgiven for the sake of being short and concise, right? But the more correct one is DA, obviously. So, so this will be the deoxyadenylate, and or you can also say deoxyadenosine monophosphate, right? So deoxyadenylate or deoxyadenosine monophosphate. Now this one is guanine base. Guanine base, so you can say deoxyguanylate or deoxyguanosine 5' monophosphate. Again, DG, DGMP. Deoxythymidylate. Now this is a pyrimidine base, right? Or you can say deoxythymidine 5' monophosphate. So it will be DT, DTMP. And finally, the 4th component, the second pyrimidine base in DNA, is based on a nitrogenous based cytosine. And so you can say that this is deoxycytidylate or deoxycytidine monophosphate. So DC, DCMP. Yeah, so the nomenclature is a little, I wouldn't say it's confusing. It's just that you have to go over this multiple times. And that's why I said to you that every time I teach this class, actually, I have to go over this myself before I actually teach this lecture to remind myself what these things are called. So. All right, so these are ribonucleic acids. So these are components of RNA. So again, you need to know the structures, names, and symbols. So obviously there is a mistake here. Can you find a mistake in this sentence?
It says 4-letter codes, but down at the bottom they look pretty 3-letter.
So where's the mistake? So where's the mistake? There are two mistakes in this sentence. Find those for me.
They're not deoxy anymore, so it's not a
Right.
D.
Yeah, the mistake obviously came from copying and pasting. Be careful with that yourself, right? So obviously there's no D here, and there is no D here, right? So the ribose is not deoxyribose. It's just a normal ribose with the hydroxy group. All right. Do I need to go over this again? All right, adenylate. Now you can see this is not deoxyadenylate, right? And this is adenosine monophosphate. So A and AMP. And nucleoside is adenosine. G, GMP, guanosine. U, now the difference is the second difference in between the two slides is that not only we're removing the D's, we replace T, right? So here's T. We replace T with U. Thymine with uracil. Or in terms of nucleotides, we replace thymidylate with uridylate. And finally, cytidylate. Cytidylate, so this will be cytidine monophosphate, C, CMP. And the name of the nucleoside is cytidine. Okay, so this clarifies things a little bit in terms of nomenclature. So I think the table should be very useful, right? So keep looking at this table a couple of times a day. Eventually it'll stick. And what else do we need to talk about? Let's see, let's look at the glycosidic bond. Let's look at the glycosidic bond. So in nucleotides, then the pentose ring is attached to the nitrogenous base via the N-glycosidic bond. So so if this is the ribose, right? So let's say this is pyrimidine, right? And so in pyrimidine, let's make this uracil. Let's make this uracil. like so, and like so. Whenever you draw uracil, uridine as a nucleoside, always draw the 2' hydroxyl, right? Do not get confused. In uridine, there is no, there is always 2' hydroxyl because it's part of the RNA. All right, so this is the glycosidic bond. And remember the numbering system we're utilizing. This is 1, 2, 3, 4, 5, 6. So, so this is the glycosidic bond. So this is the anomeric carbon right here, right? And so this is beta configuration. We already decided it's beta. And it's a really bad beta. Beta. Okay. And so we start numbering on nitrogen 1. So the uracil will be attached to the ribose ring through nitrogen 1. Now the bond is stable towards hydrolysis, right? Because now it's not hemiacetal. Remember, it's a full acetal. Even though the difference between carbohydrates and nucleosides or nucleotides is that one is the carbon-oxygen bond, but the other is a carbon-nitrogen bond. So this is, sometimes people call this mixed acetal. It's like COCN acetal. So one is oxygen, the other is nitrogen. But it's, in general, it's quite stable as well towards hydrolysis. And it's very important, right? It's very important that it's stable, because if it wasn't stable towards hydrolysis, then our DNA would be, or RNA, would be breaking down all the time, introducing all sorts of genetic mutations. Okay, and let's do the same thing for purines. So let me do a purine. See if I can draw one without looking at anything, without cheating. I'm gonna do the adenine. I'm gonna do the adenine. Now remember, this can be deoxy or can be oxy. I'll do the deoxy one. I'll do the deoxy one. So here I'm gonna start, so this will be nitrogen 9. Like so. Carbon nitrogen double bond. NH2. Like so. Okay. So so this was this was uridine, right? And this was deoxyadenine. Deoxyadenine. And so numbering, remember, we're gonna start at this position. 1, 2, 3, 4, 5, 6, and then 7, 8, and 9. All right. So in pyrimidines, it's going to be position N1. In purines, it's going to be position N9, right, on the nitrogenous base. So stable towards hydrolysis, especially in pyrimidines. Can you tell me, let's look at this from the point of view of organic chemistry. Why do you think pyrimidine, let's say drawn here, is more stable towards hydrolysis than purine? So in the hydrolysis, what will happen is, I'm going to draw the mechanism for you. Basically, what you will do, you will protonate this your nitrogenous base, right? You will protonate the nitrogenous base, H+. You will protonate that, and then you get a positive charge here, right? And then water will come in. And we'll attack that. And you get the OH. So that's the hydrolysis. Through hydrolysis, you lose the nitrogenous base, and obviously you lose the genetic code, right? So why are purines more susceptible to hydrolysis? What do you think?
Is it just because there's more places for hydrogens to interact on there?
Yeah, I think that's a good answer. I mean, there are a number of answers one can come up with, but I think that's a good one. So also keep in mind that actually in this pyrimidine, particularly in uracil, this lone pair here on the nitrogen is not very basic because it's delocalized on the carbonyl groups, right? Remember structures of amides? Amides are not very basic because the lone pairs on the nitrogen are not very accessible. Whereas here you have this lone pair, this lone pair, this lone pair, and more importantly, even this lone pair. In fact, this is the most important one on the N7. And so yeah, so purines are more susceptible to hydrolysis because of this possibility with interacting with acid or just simply hydrogen bonding with a solvent, right? But both cleavage are catalyzed, both reactions are catalyzed by acid. Well, I think now there's something important on the next slide. I would like to have more time to discuss it. So I think we're gonna stop here. So we already have, it's 12:15. So do you guys have any questions about anything today? So we have 2 more lectures on this material. 2 more lectures and then the next test. All right, well, let me know if you have any questions, and have the rest of the week. And
I have one more question before.
Yeah.
Are you going to post like practice problems again like you did before?
I don't know why. I mean, you know, the test is going to be similar. I decided I'm going to reduce number of questions to 25.
Okay.
30 to 25, but the test is going to be very similar. So from the feedback I got from students, majority of students like that.
Okay.
And the majority of students really like the fact that it's hard to cheat on this test. And it seemed to be that was the most important thing for students, to make sure that students don't cheat. So.
Okay, thank you.
Mm-hmm. Okay, sounds good. All right, I'll see you next Monday.